Welcome to AP Biology. This video is an introduction to cell division and the cell cycle. Cell division is referred to as mitosis, and the key thing to understand about mitosis is that it produces exact clones of the original cell. The original cell is called the parent cell, and the parent cell is going to divide and form two daughter cells. Each daughter cell is an exact replica of the parent cell. There are three different functions for cell division or mitosis. Unicellular organisms actually reproduce through the process of mitosis. Since they reproduce through mitosis, they are producing exact clones of themselves. A good example of an organism that reproduces through mitosis is bacteria. Since bacteria reproduce through mitosis, they need to have a high rate of mutation in order to produce genetic variation. Human beings use mitosis for these two reasons. One is going to be growth and development, and two is going to be to replace dead or old or worn out cells within the body. Living organisms grow by getting more cells, not by getting larger cells. When the sperm fertilizes an egg, it creates a zygote. Each of us was a zygote for about 30 minutes. And after 30 minutes, that zygote goes through its first round of mitosis. The parent cell divides and it forms two daughter cells, which is exactly what you see right here. Those two cells will then divide and form 8. 8 will divide and form 16. 16 will divide and form 32, so on and so forth, until you create a large multicellular organism. We also undergo mitosis to replace dead or old or worn out cells within the body. Your skin is completely replaced about every 30 days. And that's because the top layer of skin is constantly flaking off. Your skin cells are constantly replacing themselves through the process of mitosis. Every cell in your body contains your entire genome. Your genome is the collection of all of your genes, all of your sequences of DNA. Together, the genome is made up of around 23,000 genes, and all 23,000 genes can be found in every single cell within your body. If we were to unwrap all your DNA, it would measure out to be about 9 feet or 3 meters long. Before a cell can divide, the entire genome needs to be copied. It needs to be copied so that each daughter cell gets a complete copy of that genome. Your entire genome is found within the nucleus of your cells. Within the nucleus, the DNA is packaged into structures called chromosomes. Every species has a unique number of chromosomes. Human body cells contain 46 chromosomes, where if you were to look at a body cell of, let's say, a chimpanzee, they have 48 chromosomes. Our normal body cells are referred to as somatic cells. Somatic cells have 46 chromosomes total, or they have 23 pairs of chromosomes. And the fact that they have pairs should make some sense to you because you inherit one set from your mom and you inherit a second set from your dad. So you get 23 chromosomes from mom, you get 23 chromosomes from dad. The only cells within your body that do not contain 46 chromosomes are your gametes. Gametes are either sperm or egg. Gametes contain half the number of chromosomes, or they contain 23 chromosomes. If you look at this picture here, this is a picture of a karyotype. A karyotype is simply just a picture of all 23 pairs of chromosomes, or all 46 chromosomes total. In this slide we go over the structure of a chromosome. It's better to understand it using the picture. Chromosomes are made up of DNA, wound around proteins called histones. Each of these red balls that you see here is a histone. Histones are proteins. The purpose of having the histones is to condense the DNA. The DNA gets wrapped around the histones. It's like wrapping string around a ball. And by wrapping string around a ball, you're able to fit a lot of string or a lot of DNA into a really small space. When the DNA gets wrapped around the histone, together the DNA plus the histone create a structure called the nucleosome. When all these nucleosomes are put together, this structure that you see right here is referred to as chromatin, or we can call it a chromatin fiber. Chromatin is very loose, it's very open, and it's the material that our chromosomes are composed of. Normally within the cell, when the cell is not physically going through mitosis, the DNA can be found in the cell or in the nucleus in this form of loose open chromatin that we see right here. It's important that chromatin is loose and open because it gives the cell access to the DNA. So the cell could use the DNA to make all of its proper proteins. When a cell is undergoing the process of mitosis, the chromatin is going to condense and it's going to form these structures that we call mitotic chromosomes. It is only during mitosis that we can actually see the chromosomes under a microscope. The reason why we can see them is because they're much more condensed. Here we can see how the chromosomes, or how the chromatin, gets condensed. You can see that the chromatin is looped, and then those loops are looped, and all those loops are packaged together to make this really condensed structure that we call a mitotic chromosome. Now, when we have a mitotic chromosome, there's no access to that DNA. That DNA is not being used to make proteins. It's only going to be used during the process of mitosis, or when the cell is dividing. Here we see another picture. All the way on the left here, this is a picture of that loose open chromatin. We call it a 30 nanometer chromatin fiber. And again, this is how the DNA is normally going to be found in the nucleus anytime the cell is not dividing. Each of these purple balls that we see right here, these are all the histones. And the blue string is the DNA that's wrapped around these histones. And you can see in this picture that the DNA is accessible. We could read that DNA and use that DNA to make proteins. Only during mitosis is this loose open chromatin going to be looped around and condensed to form these super compact mitotic chromosomes. And these mitotic chromosomes are then visible under a microscope. Here we see a picture of the cell cycle. The cell cycle is made up of all the events in a cell's life from the moment it was created through mitosis until the moment that it divides itself. Any time that a cell is not in mitosis, is not physically dividing, the cell is in interphase. Interphase composes a bulk of that cell's life, and during interphase, that DNA is just going to be found in the loose open chromatin. Interphase is made up of three smaller phases. You have G1, you have S, and you have G2. The G stands for growth, and during G1 the cell grows, and more importantly, during G1 the cell is performing all of its normal processes. It's doing whatever it is intended to do. During the S phase, this is when the cell really begins preparing for mitosis. During the S phase, all the DNA replicates itself. So you go from having 23,000 genes within that nucleus to now having 46,000 genes within that nucleus. During G2, we also see cell growth, and the rest of the cell is preparing for division. All the other organelles replicate themselves. After G2, if conditions are right, the cell will leave interphase and it will enter the mitotic phase. It will enter mitosis. Mitosis is made up of two smaller phases. Number one is mitosis itself. And mitosis is technically when the nucleus divides. Following mitosis, we have cytokinesis. Cytokinesis is when the cytoplasm, or the rest of the cell, divides. During mitosis, that one parent cell is going to become two daughter cells. And each of those daughter cells is an exact replica or a clone of the original parental cell. Here we see what is going to take place during the S phase. If we look at an original cell in this picture, let's say that this cell is in G1, and this cell right here has one chromosome inside of it. Our cells would all have 46. We begin by having just a normal single chromosome. Well, during the S phase, all the DNA replicates itself, and this chromosome makes an exact copy of itself. And these two copies are held together by a structure called the centromeres. Each of these copies is referred to as a sister chromatid. This is a sister chromatid, this is a sister chromatid. This entire structure right here is referred to as a duplicated chromosome. That duplicated chromosome is made up of two sister chromatids, but it is considered one chromosome. So this could be a little confusing. This thing you see right here, that's one chromosome. This entire thing right here, that is also one chromosome. It's just one duplicated chromosome made up of two sister chromatids. During mitosis, when the nucleus divides, the sister chromatids are going to split apart. When those sister chromatids split apart, each chromatid becomes its own individual chromosome. So it's important to understand that during mitosis, the chromosome number stays the same. We began here with a cell with one chromosome. We end with two daughter cells that each has one chromosome. In our cells, you begin with a parent cell that has 46 chromosomes, and you end with two daughter cells that each have 46 chromosomes. Here you see what's happened to the amount of DNA. Right, here's that original cell during G1. You have single chromosome, and you have this single piece of DNA. During the S phase, the DNA replicates itself, and we get a duplicated chromosome. You can see that that duplicated chromosome has twice the amount of DNA. These sister chromatids are going to separate during mitosis. Each daughter cell gets one of those sister chromatids. When those sister chromatids separate, they're each now considered their own chromosome. So we began with a cell that had one piece of DNA here. We ended with two cells that each had one piece of DNA. The amount of DNA, or the amount of chromosomes, remains the same during mitosis. This concludes this video.